The goal of Nirenberg and Matthaei's and Nirenberg and Leder's experiments was to determine the genetic code and what specific RNA sequences coded for specific amino acids. Nirenberg and Matthaei created an RNA molecule that contained only uracils. This is a poly-U RNA. They then added the poly-U RNA to 20 different test tubes that contained all 20 amino acids and cell-free extract. In each test tube, a different amino acid had a radioactive label. They then looked to see if any of the test tubes contained a radioactive protein. This would show what the codon UUU codes for. Only one radioactive protein was found, and that was phenylalanine. This showed that UUU codes for phenylalanine. They then did the same process with CCC, AAA, and GGG. They found that CCC codes for proline, AAA codes for lysine, and GGG did not code for anything. A few years later, Nirenberg and Leder created an experiment to determine what amino acids the rest of the codons code for. They started by mixing together a trinucleotide, a ribosome, and 20 charged tRNAs. They had 20 different test tubes with each one having a different amino acid containing a radioactive label. They allowed binding between the components to occur and then filtered out unbound amino acids. If the filter paper was radioactive, it showed that the trinucleotide that was present codes for the amino acid that was radioactive. For example, when the trinucleotide GUU was added, valine was bound to the filter paper, and it was radioactive. This showed that the codon GUU codes for valine. They were able to determine 61 different codons using this process.